Good afternoon and welcome to the Interoception and Bidirectional Interaction between the Peripheral and Central Nervous System session. I'm Manda Keller-Ross. I'm an assistant professor at the University of Minnesota here in the Department of Rehabilitation Medicine. My research focuses primarily on autonomic dysregulation and cardiovascular diseases, and I am mostly focused on women's health. I'm also really interested in novel therapeutic strategies to mitigate and treat cardiovascular disease risk. So we have a really exciting session here today with 3 expert speakers on interoception, autonomic nervous function, and neuromodulation. I'm going to start by giving a background on interoception. This is not a new term. It's been around for many years, but it might be a bit of a new term for some of you. So interoception refers to the representation of an organism's internal state and includes the processes by which it senses, interprets, integrates, and regulates signals from within itself. And this is a definition from Dr. Chen's paper. I didn't quote it just because you were here, Dr. Chen. I actually read this paper last week and thought, this is brilliant. This is perfect for my talk, and then you came. I swear that's true. So it's our nervous system's ability to represent our own internal world. So it's an interaction of these networks, particularly afferents, or sensory information to modulate basic physiological processes. So it can modulate respiration, blood pressure, motor responses. So the concept of interoception dates back to about 150 years ago, to Claude Bernard, okay? And he coined this term, milieu intérieur, and I'm not French, so I'm not going to say that correctly. Claude was a French physiologist, and his idea was that there was an having a hard time speaking today, identifiable set of physiological parameters that defined the normal internal state of the organism. He first described the concept of the internal environment, which we now know as homeostasis. And then we have Dr. Pavlov, who's a Russian scientist, and we know his studies on classical conditioning, where we have the conditioned response of the salivation of the dog by a ringing of a bell. Dr. Cannon was one of the pioneers in the study of autonomic nervous system, isolating nerve terminals and pointing out the role of sympathetic as a mediator of impulses between nerve and muscle. The first known usage of the concept of interoception is 1906 by Dr. Charles Sherrington in his book, The Integrative Action of the Nervous System. So it's not that these terms are used interchangeably, but without reliable afferent information traveling from the body to the nervous system, it would not be possible to control or maintain this homeostatic milieu. So we're going to hear a lot about afferent activity. And if you look at the left side of this slide, you'll see peripheral organs with spinal afferents. So this is the idea that organs have sensory afferents coming from the organ to the spinal cord, through the dorsal horn of the spinal cord, back up into the brainstem. On the right side, we see the cranial nerve 10, otherwise known as the vagus nerve. Okay, we've heard a lot about the vagus nerve today. And so we have the same concept here. We have lots of afferent information coming from the organs to communicate to the brain. And in the center here, you see the brainstem, pons, and medulla. And one of the major central integration centers is the nucleus tractus solitarius, or the NTS. And so there are other structures in the brain that also play a role in this. But this is where the communication happens in order for us to regulate autonomic function. And so we're going to be hearing a lot about that today. We're first going to hear from Dr. David Darrow. And he is going to be primarily speaking about modulating at the spinal cord level. And so his talk will be on spinal cord stimulation and how to restore autonomic function after a spinal cord injury. And I will introduce him in just a second. Our next speaker will be Dr. Maya Keelberer from Duke University. And she is going to take a deeper look into the gut. And we're going to hear about the neuropods and the neuropod cells. It's a sensory cell in the gut surface and how it communicates directly and rapidly with the brain. And then we're going to hear from Dr. Jeanie Park at Emory University. And she's going to talk about how we can leverage interoceptive pathways to improve sympathetic regulation in chronic disease. And so I circled lots of different organs for her because she's going to be talk about modulating sympathetic activity and how we can do that by breathing. So I first want to introduce Dr. Darrow. Dr. Darrow is an assistant professor in the Department of Neurosurgery here at the University of Minnesota and the Roxwald Kaplan Endowed Chair for Traumatic Brain Injury at Hennepin County Medical Center. He specializes in functional and pain neurosurgery. Dr. Darrow treats disorders, diseases of the central nervous system, and neuromodulation, including epilepsy, movement disorders, trigeminal neuralgia, chronic pain, and psychiatric diseases. So please help me in welcoming Dr. Darrow.
Dr. Keller-Ross is very persuasive. Somehow she convinced me I was expert enough to talk about interoception right after lunch, when everybody's interoception is essentially saying it is nap time. So I'm David Darrow. I am a functional neurosurgeon here at the University of Minnesota, and I wear a few hats. But today I was asked to talk about spinal cord stimulation to restore function, specifically autonomic function, really through the lens of interoception. And I feel like interoception is one of those holy grails where we're all trying to learn more about it, but the idea has really been around for a long time. Now, it's important for me to tell you my biases. I get research funding from Abbott Labs. I have my own intellectual property from the work that I do, none of which I will talk about today. But this is an off-label use of a medical device. This is not FDA approved, and this is in a clinical trial. First, it's important for you to know what I do. I am a pain and functional neurosurgeon, and my clinical practice is mostly focused on putting electrodes into people's brains and spinal cord. And generally, these electrodes allowed me to record and to stimulate in the hopes that we're able to modulate its function to hopefully for the better. And I have a joint lab with a psychiatrist here, and we study the relationship between the electrical function of the brain, how we perceive the world, and how that informs the decisions that we make across several diseases. And there's a lot of people that go into this, that support this research, and it's important for me to, show them up front. We have a really amazing team. And I also simultaneously run a lab focused on electrophysiology and modulation to restore function after traumatic spinal cord injuries and brain injuries at the county hospital. And we also have a really great team that overlaps significantly with all my collaborators and students across, from the people on the ground to the engineering teams and the postdocs. It's just a tremendous amount of work, and it's important you know that. So today I was asked to come at restoration of function through the lens of interoception, and I think William James really said it, Dr. William James really said it best. I said I should consider these emotions only so far as they have bodily movements of some sort for their accompaniments. But my first point is to show that their bodily accompaniments are much more far-reaching and complicated than we ordinarily suppose. So it's a simple idea with a very complicated underlying foundation. And I want you to think back to that last time that you felt just overwhelmingly hot. And I know that's very difficult for us to imagine here in Minnesota. It was last week. We're all taught in elementary school the 5 senses, right? But were any of these senses the ones that you used to determine that you felt hot? Could you see that your body was hot, or hear, or smell, or taste, or, what was it? How did you know that you were overwhelmingly hot? Our mind's representation of this state of our body has vexed scientists for as long as science has existed and before. And if we contrast this with what our five senses usually do, you quickly realize that feeling unwell or feeling hot or cold comes with an emotional and motivational feature that tends to motivate us to change our state. It is inherent in this type of feeling. And as a result, this is an essential homeostatic mechanism of our body. The dialogue that occurs back and forth between the mind and the body about the homeostasis of our body is interoception. We try to simplify it and say it's just the afferents, but the afferents cannot exist without the efferents. It is one larger process going on. Sensations like pain, temperature, and our visceral bodily feelings are different from touch due to this affective, motivational aspect. In particular, the motivational and emotional aspect of, for example, thermal sensation is particularly underappreciated. When you're hot and sweaty outside and you finally grab that cold glass of water, it feels good, right? When you're here in Minnesota and it's freezing outside and you finally get to warm up by the fire, it actually feels good. It is quite rare that you sit down at your computer and put your hands on your keyboard or your mouse and it feels good. It just doesn't. These are different things. We're talking about different things, but they use a lot of the same infrastructure. And what has become more clear over the past 100 years is that these sensations are mediated through different pathways of the body. And the lines between mind and body become increasingly blurry as we appreciate interoception as this constant, vigilant, bidirectional dialogue amongst the entire nervous system, which is ever present throughout our body. Most of us know about the outgoing portions of the autonomic system, the efferent portions of the autonomic system. But many may not appreciate the afferent sensory autonomic system. And I know it's been touched on, but just one more time, there are generally two major ascending peripheral or afferent pathways that transmit interoceptive signals to the central nervous system. Signals in these two pathways relay through two distinct types of peripheral ganglia. Ganglia residing in the craniovagal pathways, such as the nodose or jugular ganglia, often project to the nucleus tractus solitarius of the brainstem, whereas the dorsal root ganglion from all the segments of along our spinal cord, they project information through the spinal cord itself, like the cortical spinal tract. Visceral afferents that travel along the cranial nerves, including the vagus, are referred to as the parasympathetic system, and those that travel through the spinal thalamic tract of the spinal cord are often called sympathetic afferents, though it's still a little bit controversial. And it's hypothesized that generally the vagal afferents are carrying a lot of the mechano and sensory signals, whereas the spinal afferents, it's temperature, pain, and tissue injury. From there, these signals are projected. From there, these signals are projected through the thalamus and the insula and into the amygdala for higher order processing. And we know even in our own lab, when we're recording brain activity from patients with epilepsy and we're trying to localize the seizures, we take that opportunity to have them do a very simple virtual task. And when this happens, we see this task allows you to either chase a prey or have predators chase you, or sometimes both. And the result of that is a significant change in brain activity here in the spectrogram in the amygdala. Whenever we're put in situations that would normally evoke our flight response, such as a predator bearing down on you, this is what keeps you safe. And this is the same system that most of us take for granted that is significantly disrupted in spinal cord injury. And I think everybody is familiar with spinal cord injury, the motor aspect, the loss of motor function after spinal cord injury. But spinal cord injury is so much more than that. First, it's important to know the basics. The incidence in the US is about 54 cases per million, and this means 18,000 new cases of spinal cord injury per year. And there's about 300,000 people living with spinal cord injury currently in the US. And they're primarily, historically, were due to motor vehicle accidents, falls, and violence, which is on the rise. And about 59% of them are cervical injuries, and that leads to tetraparesis or tetraplegia. And about 41% is thoracic spinal cord injury, which leads to paraplegia or paraparesis. Spinal cord injury can tell us a lot about interoception because the primary disruption is to the sympathetic efferents and sometimes afferents, as well as the lower sympathetic efferents and afferents that go to the bladder and pelvic organs. We see this significant, oh yeah, here's the more powerful one. I'm not going to point to all the screens, so just, We see, significant disruption in this, in especially this efferent system, controlling the blood vessels, the heart, the gastrointestinal system, the bladder, essentially everything that you have in your peritoneum and throughout your body. And as a result, you can predict that many patients with spinal cord injury suffer from cardiovascular dysfunction, problems with even breathing, gastrointestinal function, urinary function, sexual function, and even things like pseudomotor activity, sweating, and thermoregulation. But these symptoms vary based on how severe the injury was and what level the injury was at. So you can learn just a tremendous amount. But what many people don't know is that there's even burgeoning literature that spinal cord injury actually affects our own representation of self in certain scenarios. Here in Asia A spinal cord injury patients, meaning they have no motor or sensory function below the level of the lesion, and these are all cervical patients, they were compared with able-bodied controls to identify emotions present on people's faces and the emotions that are generated from pictures of different environments. And there were no differences found in interpreting the emotions on people's faces. That was easy. But there were significant differences identifying the emotions paired with dangerous and fearful environments as well as hostile or angry environments. And this indicates that even with just a simple spinal cord injury, disruptions to this bidirectional homeostatic system of the body simultaneously affects the brain. And they're just looking at images, right? How is this occurring? Maybe William James was right. But there have been some breakthroughs for treating spinal cord injury that have evolved over the past 20 years. And we here at the University of Minnesota and Hennepin County have been testing them for the past 6 years. And we've been attempting to use tonic stimulation, very simple stimulation, to restore function for those with chronic motor complete thoracic spinal cord injury. And we see a range of lesions of the spinal cord, from nearly, intact looking ones to completely decimated ones with lots of area of stroke. These are all mid-sagittal plane MRIs showing you the best slice we have of the spinal cord. And the theory behind how tonic spinal cord stimulation works is that we're stimulating below the level of the lesion to restore a more normal balance of excitatory and inhibitory fibers. And what that does is it makes it possible for any of the remaining supraspinal tracts, the ones coming from the brain, to actually resume control. Because maybe there is 1 or 5% of tracts remaining through these fibers. So if we just stimulate the area that we want to restore, now, because there's a normal balance of excitation and inhibition, they can work. In a crude sense, this is kind of like amplifying what's left. And even on the first day after surgery, for these patients 6 years after injury and 8 years after injury, we simply just turn it on. This is the first time. And they have not moved in 6 and 8 years, and they have no spasticity. We ask them to turn it on, and immediately we see, dramatic results. And what's more interesting to us is after many months of nearly continuous stimulation, a significant portion of the patients start developing the ability to move, albeit less, without stimulation on at all. There's no stimulation on, and this gentleman has been paralyzed for 17 years. Still, no spasticity. And while it is far from complete restoration, these movements are substantial when compared to their previous history, which was no movement at all, or just a little bit of spasticity. And now we've shown in just a recent upcoming publication, I think that's out this month, that we're able to restore enough movement to restore the ability to cycle. This is a cycle that pedals itself, but as you can see when stimulation on, it actually can be reversed and pedaled stronger. And what we found is that just stimulation itself is not sufficient. In order for one of these participants to actively pedal, it requires both stimulation and effort. Here you can see without effort, stimulation does not provide any assistance. But as soon as effort and stimulation are applied, we see a significant change in the max amount of power that you can generate on this cycle. Hinting again that there's a big volitional component to this. And interestingly, none of the demographic or medical information about these patients explains any of the variability that we see in their ability to cycle, whether it's age or years from injury or the picture of their spinal cord above and below the lesion. But it's important to remember that despite restored motor movements, almost all of these patients are sensory complete. They have no motor or sensory function, and they move without proprioception or sensation. And we already know that if you do not have proprioception or sensation, it is almost impossible to walk, separate from spinal cord injury. It's also important to remember in these patients without sensation that there are no signals getting to the brain to tell them that something is wrong. Instead, oftentimes patients find out because they feel unwell from the systemic side effects of unregulated blood pressure, for example. It's sort of an indirect method of interoception. Instead of feeling the heat from the heater that's burning your leg, you feel unwell because your blood pressure is now 200 over 100. And restoring autonomic function in spinal cord injury is perhaps of much higher importance because many patients have numerous issues that are ongoing that they would love to have some benefit, including sexual function, bowel bladder function, blood pressure. And we have now demonstrated with our collaborators that spinal cord stimulation improves the typical resting hypotension associated with higher injuries. And even when challenged during tilt table testing, spinal cord stimulation can reverse autonomic dysreflexia. Here on the top, we've tilted them. We turn on a sham stimulation, nothing happens. We turn on targeted stimulation, and we see a significant rise in this systolic blood pressure. Here, we're simultaneously improving cerebral blood flow. And lastly, we improve cardiac contractility on the fly. But for patients like this person on the left, who has no problems with their cardiovascular system because the injury is much lower, stimulation doesn't seem to do anything. So we have, importantly, carefully analyzed all of the blood pressure and EKG that we see in our trial from spinal cord stimulation, and we don't see that it worsens arrhythmias. We don't see that it's really a pain response that's causing unmitigated sympathetic activation. We don't see dangerous increases in blood pressure. What we do see is that actually it's better heart rates, better arrhythmias, and better blood pressures when the stimulation is on. So there is hope that neuromodulation can provide a method of restoring some critical autonomic function after chronic spinal cord injury. But does this translate to a change in interoception? In these early days, we still don't know, but we are seeing changes that are hard to explain otherwise. To our great surprise, some patients in our study started reporting improvements in their sexual function. And we've just recently published our first report in women. As you might imagine, we were not prepared for reporting all these findings, but we have done our best to catch up. And now we see reductions in sexual distress, improvements in overall sexual function, improvements in arousal, which is generally surprising for any therapy for spinal cord injury. And we think it is plausible that by providing more balance to the sympathetic and parasympathetic homeostatic system through neuromodulation, we may be secondarily partially restoring interoception. We constantly hear reports that we were not prepared to quantify and are trying more and more to do so, of patients telling us, I just feel so much better with the stimulation on. And, we ask them, do you want to elaborate on that? And they're like, no, it is what it is. I'm just telling you my report, which is the challenge of interoception. It's easy for us to weigh in, but it's so hard to measure and to quantify. They also say things like, I can be at work longer throughout the day. I don't feel fatigued by the end of the day. I don't have a hard time thinking. I don't feel like I have a headache towards the end of the day, which, we imagine is due to improvements in blood pressure. Or I can tell now when I need to go to the bathroom. I can't explain that, but they tell me this. Or now when I'm in the heat, my legs are sweating constantly. In addition, I hear things like, why do I have to go to the bathroom more than twice a day? I used to schedule it, for 2 times a day. And my response was like, most of us go to the bathroom more than twice a day because we sense our bladders full. So I think we can start learning a lot from these patients, especially because they really know what's going on and can tell us a lot about their own interoception. So I think our learning points are the interoceptive perspective remains really critical for understanding how visceral and autonomic treatments may be contributing to overall well-being. And we should incorporate assessments that continue to be systematic in our research. Second, that bidirectional homeostatic control system of the autonomic nervous system involved in interoception is really now, I think, the standard model for us to improve upon. Third is that there is hope that neuromodulation may provide a means to tap into novel treatments for improving well-being, as we've all have seen today from many of the other speakers. And lastly, we can potentially make the argument that these are perhaps more important than motor function for many of the patients struggling or suffering from chronic spinal cord injury. Thank you so much. Thank you to our funders. We do have a postdoc position open, and feel free to email me at any time. Thank you so much.
We have some time for questions.
David, great talk right here. So where do you think you're stimulating, and do you think there's any effects that could be relayed to reestablishing any neural connections?
That's a great question. I'm pretty pessimistic. So my answer to you is that I think we're stimulating some of the sympathetic ganglia in the spinal cord, and we're providing a new basal amount of tone. And the dynamics, my hope is that the dynamics, the interplay between the parasympathetic and sympathetic system are somewhat restored. Now, it's hard for me to explain these long-term changes, but I have been looking for long-term changes with the blood pressure, and I haven't seen them yet. So for motor, I see some things that make me think, maybe there's something going on. But on the blood pressure side, I haven't seen it yet, which, is not necessarily a bad thing. It just means they still need stimulation, and that seems to be still. But, if we could get people like Dr. Keelgaard over here to put in some other electrodes or allow us to put in some other electrodes, then maybe there is that possibility that we could be doing some rewiring.
So when people do evoke spinal cord action potentials, they typically see that at normal amplitudes, they're only activating A betas and not activating A deltas and C fibers, which are more canonically associated with sympathetic afferent input. I'm just curious in terms of, what do you think is being stimulated below the lesion that kind of creates this change in autonomic function?
That's a great question. So you're absolutely right. There's preferential activation of the larger fiber, There's two things to consider here. One is that in these patients, they have constant segmental plasticity that is ramping up things like spasticity. So their system is a lot more sensitive to stimulation than perhaps someone that has normal cortical suppression, that comes, from above. The second is that generally speaking, the stimulation that we're doing is higher than we would for example, chronic pain. And so we stimulate, not always, but there's a much higher, on average, we stimulate much higher. So we may be recruiting, some other fibers. That being said, we do not see the typical sympathetic responses due to noxious stimuli that patients would if they stub their toe or they're burning their leg. We don't see that ever, because then you see a lot more ringing of blood pressure, through the autonomic system, of blood pressure skyrocketing and things like that. So I think we're still stimulating the bigger fibers, A beta, but the secondary effects is that is translating to downstream effects on at least the local motor side through those interneurons. And if you believe people like Cortine, they're the V2 neurons that are primarily meeting that. Yeah, but that's a great question. Thank you.
We have time for one more quick question.
So
you can't pick Alec.
it's a nice one, I swear. No, right? Especially since you name check Michael, I'm looking at this and I'm thinking about everything he presented this morning about how actually we see these same autonomic dysfunctions in patients with trauma, depression, chronic pain. And so I got to ask, do you think you'd get the same effects in somebody who wasn't spinal cord transected, contused, who was spinal cord intact?
I don't, because, there's some caveats there, but overall, I don't. And the reason is I think many in the field, and I'm not one of those people, there are many people that have been around for a long time that have been trying to use spinal cord stimulation to modulate the autonomic nervous system, and it is hard. It is hard. I get to cheat because the patients I have don't really have much control of their autonomic system, and it's easy to perturb the system. I think that ordinarily, there may be some caveats to this. There are some specific examples where it may work, but I think overall, I don't think it's low-hanging fruit. But don't give away any of our other grants right now, Alex. So
Thank you, Dr.
Thank
Darrow.
you.
Okay, our next speaker is Dr. Maya Kaelberer. She is a sensory neurobiologist from Duke University. Dr. Kaelberer was one of the first to use transcriptomics to study specific populations of vagal neurons. Her research focuses on uncovering how the gut communicates sensory signals from nutrients to the brain. In 2018, she was the leading author on an article in Science showing the neural basis of a new sense, a gut sense, which is what we'll hear about today. Her research seeks to uncover the secrets of how the gut and brain talk in real time. Please join me in welcoming Dr. Kaelberer.
Thank you so much to the organizers, and thank you so much for having me here to share my research and my work with you. As was mentioned, I am a sensory neurobiologist, and what really excites me about sensory neurobiology is kind of this concept. I'm sure that you've heard this phrase, I think, therefore I am. As a sensory neurobiologist, however, what I actually believe is I perceive, therefore I am. And what I mean by that is our senses and how we're sensing the physical world and turning it into an electrical impulse is really what's driving our cognitive mind. And some of the best examples of this actually come from the visual field. So you're probably looking at this and thinking, how come I can't focus on that black dot? It just keeps moving around. Well, the black dot does not exist. This is actually a result of the early sensory processing within your retina. And this is a great example of how our visual system isn't actually communicating the reality of the light, but it's actually just filtering what it thinks is important and what it has developed to think is important. So what does that mean for something like the vagus nerve? And so we've all heard about the vagus nerve. I like it because it's called the wandering nerve, and clearly it has wandered into all of our lives, or we would not be here today. But the vagus nerve innervates our gut, our heart, our lungs, our whole viscera. It innervates this area where we know perception is there, as our previous speaker had pointed out, but we're not really able to communicate what does that mean. And so I study the gut-brain connection. And so what I mean by the gut-brain connection, I focus on the vagus nerve and how it connects the gut to the brain. So to orient you, within the gut, we have a series of villus. Within a single villus, there's a sheet of epithelial cells. There are many different types of cells. I'm interested in this one type of cell, the neuropod cell. Now, the neuropod cell is the sensory transduction cell in the gut epithelium. It's able to take signals like mechanical, thermal, or chemical signals like nutrients. It senses them, and then it quickly synapses with vagal afferents in order to communicate directly to the brain what is going on in our gut. And as was mentioned, this discovery of the circuit happened in 2018. So when this happened, the number one question that I got was, why have a gut sense? What is it about a gut sense? Like, why do we need it? I can see my food, I can smell it, I can taste it. That's where all my decisions are happening. What could the gut possibly be telling me more? And so in order to look at this, we relied on this assay that's been in the literature for a really long time. It's this two-bottle preference assay. So what we do is we give the mouse the option between two solutions. It can either have the caloric sucrose, this is just normal sugar, or it can have the artificial sweetener sucralose. And when you give it these two options, they're about isosweet, the mouse will always choose sucrose over the artificial sweetener sucralose. And this happens independent of taste in the mouth. So there's something going on in this animal that it knows which one is the caloric sugar and which one is the non-caloric sweetener. And so in our laboratory, we use vagal nerve electrophysiology in order to record, well, what are the possible signals from the gut that the vagus nerve is even interested in? And this is our gauge of being able to tell, like, is this a signal that's important to the gut or not? And so when we break down sugar, we can break it into different components. So when we test the vagal response to sucrose here at the top, we get these increase in firing rate that you see in the orange. That'll happen when we perfuse it through the intestine. So I'm just going through the proximal duodenum. And if we look at different types of sugars, we see also the vagus response to them. So that's down here, the glucose or the galactose. Interestingly, fructose, we actually don't see a vagal response to. Then we also have sugar analogs. And I'll specifically be talking a lot about alpha MGP. This is an analog that gets transported into the cell but is not metabolized. And then we have artificial sweeteners. And so all three aspects of this sugar sensing are being communicated via the vagus nerve. And so how is this happening? Well, we can look at just the nodose neurons themselves. These are the sensory neurons of the vagus nerve. And when I wholesale patch them, there's no response. So they themselves are not sensing sugar. However, if I take out these sensory cells, these neuropod cells, and I give them access to either glucose or sucralose, what I find is that there's this breakdown of the majority of them, like the caloric sugar glucose. There's this middle population that responds to both, and there's a smaller population that just pays attention to the artificial sweetener. And so at the level of the sensory cell, it's actually able to distinguish between these two sweet stimuli. And I'm not going to go into the data for this, but what we had found is that the difference is actually the sensing transporter, so the sensory receptor on the membrane, where what we're seeing is that glucose, or real sugar, is being transported into the cell via sodium glucose transporter, whereas artificial sweetener is using the sweet taste receptor. And the sweet taste receptors is the method that we use to sense sweet on the tongue. And so are these neuropod cells the ones that are transducing it? We can see in culture that they can tell the difference, but are they actually the ones responsible for the signal we see in the vagus nerve? And to look at this, we turn to optogenetics. So now we're using light to inhibit the activity of neuropod cells. And so when we do this, here we see in the top trace, this is just our normal sucrose vagal response. However, when we optogenetically silence the cells, we completely abolish this response, indicating these neuropod cells are necessary for transducing luminal sugar onto the vagus nerve. This is true for artificial sweetener. This is also true for that sugar analog I mentioned, alpha MGP. And so we see that in the animal, they seem to be necessary. But in our laboratory, we actually have this really cool culture system. So in vitro, we can take neurons, sensory neurons, which you're seeing on the, in red on the right, and we can take our epithelial cells and we can put them in culture together. And what we see is that over time, we have time in the top right. At some point, these cells start moving around, they sense each other, and then they form this connection in vitro. And one thing about this video that I think is pretty cool is that other neuron you see is so amazed that I can recapitulate this neuroepithelial circuit. It just dies. It can't handle it. So this actually gives us the ability. I can take my sensory cell and my sensory neuron, and I can see exactly how they're communicating with each other without any of the other environmental factors. And so when I do this, the question is, okay, are they transducing glucose and sucralose? Are they both being transduced, or when they pair up, is it only one or the other? And so what I found is that the neuron is responding at the same amplitude to both stimuli. So it's not that sucralose is a diminished glucose response. They're two different signals. And then when I look at the different pairs that I patched from, we see the same thing that I'd seen in the calcium imaging, mainly that the majority of the pairs seem to be responding to the caloric glucose. The second largest population doesn't care between the two. And the third is only responding to the sucralose. So this is really showing that it depends on what sensory transduction cell is being attached to the neuron, which will determine what it responds to. And so this is now in a dish, but let's go back into the whole animal. So vagal cuff is really powerful because it kind of gives us generally what's going on. But what about on a neuron-to-neuron basis? And so in order to investigate this, again, I'm perfusing nutrients through the small intestine, but now I'm using calcium imaging in order to look at individual neurons within the nodose ganglia in vivo. And so then when I do this, I perfused sucrose and sucralose, and then tried to determine, are they the same neurons that are responding to the two different stimuli? And what I found is that they aren't. In fact, they're completely different neurons. So if the neuron is responding to sucrose, it's not going to respond to sucralose. And so at the level of the nodose ganglia, we're actually now seeing parallel pathways. And this is interesting because what it means is that at this level, at this first synapse, we're already able to go down two different routes of behavior, like what we had seen in the whole animal. All right, so what about the signaling molecule? So they're communicating to each other, but what is the neuropod cell releasing? And so we looked at glutamate. This is a main neurotransmitter in a lot of the sensory systems. And in order to investigate whether or not these neuropod cells are releasing glutamate, we use this glue sniffer protein. And if you know Lauren Luger, he comes up with the greatest names, glue sniffer being one of them. So this protein is able to detect glutamate, and then it fluoresces green. And so here now I've transfected hex cells with this glue sniffer protein, and I've co-cultured them with my neuropod cells from the small intestine. And when I add glucose, you can see that they light up. And so it's important to know that the glue sniffer does not respond to glucose on its own. So what we're seeing is that these cells, when they sense glucose, they release glutamate. So let's go back into the co-culture. Is this true in the co-culture? Can I see glutamate as the neurotransmitter? And so to do this now, I'm just activating using excitatory channel rhodopsin. I activate my neuropod cell, and then I add on kyanuric acid, which is an ionotropic glutamate receptor blocker. It completely abolishes the response. When I wash it away, the response comes back. So it seems like in this dish, what we're seeing is that, yes, glutamate happens to be this neurotransmitter. And so now, if we go back into the whole animal, is this also true with our vagal response? And what we found is that it's true. So if I now block, this is what the Ka and the AP3, those are glutamate receptor blockers. If I block just the glutamate receptor blockers, what I see is that my sucrose response is abolished. I see my alpha MGP response, so this was it gets transported in the cell and non-metabolized, is also abolished, but my sucralose stays exactly the same. And so then the question is, okay, what's the neurotransmitter for sucralose? And so we looked at another common neurotransmitter used in the tongue, which is ATP. And what we found is that that actually appears to be it. So here, when we block purinergic receptors, only the sucralose response is abolished. And so what this is showing us is that there are these two different receptors that are being used by the neuropod cell in order to detect the different caloric versus non-caloric sweeteners. And then, based on that information, it's releasing different neurotransmitters. Glutamate to say this is sucrose or this is sugar, and ATP to say this is fake news. So what about in the whole animal? Can we take this back into the behavior and test it? Well, optogenetics is really cool, and it's super powerful, and people have been using it in the brain. But in the brain, you have a skull that you can put that fiber on. And so we had to work with bioengineers at MIT in order to develop a way for us to take optogenetics into the gut. And so here on the left, we see a stiff silica fiber. And the issue with this stiff fiber is you might have noticed your gut has no real bones in there. And if you put a stiff fiber in it, it'll puncture the gut wall. And so here we have this agarose that's about the thickness and viscosity of the gut. And when we take this stiff fiber, it completely punctures through. Whereas Polina Anikiva at MIT had developed this flexible fiber optic. And this flexible fiber optic actually bends. There's a second aspect of this fiber that's actually really important for our purposes, which is that you can see that it's illuminating the length of the fiber as opposed to just the point. Well, these sensory cells, the gut is huge. These sensory cells are not contained in one dense population. And in fact, in the small intestine, they're 1 every 100 epithelial cells. In the colon, they're 1 every 1,000. And so we need to be able to illuminate a length of the intestine in order to do anything meaningful with optogenetics. So here we have this flexible fiber optic. We've implanted it in the proximal duodenum of the mouse. You see it here illuminated. And we're now actually just going to silence neuropod cells in that first 1.5 centimeters of the proximal duodenum. So again, we have our classic sugar test. We show that the control laser has no effect. This is important because these cells also have TRPV1, so they can sense heat. However, when we optogenetically silence neuropod cells, we completely abolish the preference for the sucrose over the sucralose. Now, these animals are drinking the exact same amount. They're simply no longer aware of which solution has the real sugar and which one has the artificial sweetener. And so we've basically just blinded them to know. They no longer get the information. But now we want to know, okay, is glutamate actually that signaling molecule that's telling them that this is the real sugar versus the artificial sweetener? So in this experiment, we've now implanted an intraluminal catheter, and we can now perfuse in a solution through the proximal small intestine. When we do the vehicle, of course, they still prefer their sucrose to their sucralose. However, if we now perfuse in glutamate receptor blockers, again, we see the same effect, that they're completely abolishing the preference for caloric sugar over the artificial sweetener. And so what this is showing us is that our gut sense could be actually driving our choice to eat at a level that we don't quite understand yet. And so maybe when you went out to lunch and you grabbed your bag and you started eating your sandwich and you're like, this is good, but it's not really what I was looking for. Oh, there's a cookie in here. Ah, that cookie, that's really gratifying. That's something satisfying. And that could be what our gut sense is trying to tell us. And we think at some level that it's us thinking it. But much like that dot in the circle where we think that black dot was there, it's actually our senses that are driving this choice. And so this is important for a number of reasons. Of course, you can't talk about gut without talking about obesity. But obesity, we know, is not just a one-trick pony. It's not only maybe sensing in the gut, but it has these multi-layer things. And since I'm in a bidirectional session, I wanted to talk about things that go in the other direction. So we know also that things like obesity are affected by chronic stress, for instance. And we've all just lived through a lot of chronic stress during COVID. Some of you might have gained a few pounds. Some of you might have actually eaten a lot less. But we know that chronic stress has this effect on GI function, and it can lead to a lot of severe things like irritable bowel syndrome. And we also know that chronic stress leads to this increase in uptake of palatable food. And so in this case, palatable food for these rodents is just high fat, high sugar. So comfort food. It's what we all have our own comfort food. And so I wanted to know, is there a brain-to-gut signaling pathway? Are these neuropod cells actually able to receive input from the brain in order to modulate sensory transduction at the gut epithelial? So the first thing that I need to look at then is to see, well, do they even have a postsynapse? Are they even capable, do the machinery, in order to receive input? And we had shown that they express some of these postsynaptic markers. And in the small intestine, I quantified it to be about a third. And so about a third of these neuropod cells appear to have the proteins that would allow them to receive feedback. And so what could that signal be? So we've heard a lot about the sympathetic nervous system, the parasympathetic nervous system. So I tested a bunch of candidate neurotransmitters just in culture to see what neurotransmitter could it be. So acetylcholine, this is the main parasympathetic, this is the main neurotransmitter of the vagus nerve. Norepinephrine being the main one for the sympathetic nervous system. GABA and serotonin are both major neurotransmitters within the enteric nervous system, so the gut also has its own nervous system. And then there's glutamate and buffer, and since they are a little mechanosensitive, I've added in the buffer in there. But it appears that actually, it seems that acetylcholine might be this main signal. So we're talking about the efferent vagal neurons that are potentially influencing effects on neuropod cells in the gut. And so then I wanted to know, well, can I take this into a co-culture? Can I see the efferent effect in this co-culture system? And so first, I just wanted to quantify it. Am I seeing the postsynaptic markers in my co-culture? And it turns out, yeah, they actually recapitulate what I've been seeing in the whole mouse in a similar fashion, which is what you'll notice is that the postsynaptic, the PSD95 labeled cells, neuropod cells, always have synapsin 1 in them, or almost always. And so this makes sense. There's a set of sensory neuropod cells, they're sending signals to the brain, and then a subset of those ones are also able to receive feedback down. And so this opens the opportunity for the efferent to actually affect how the sensing is now going afferently. All right, so can I stimulate activity? Because I've only shown you markers so far. And so in this experiment, what I'm doing is I'm now electrically stimulating the neuron, and I've expressed a calcium indicator in the connected neuropod cell. And so over here, you're going to see a video of this neuropod cell that I'm imaging, and then down along here, you'll see the trace. At about 15 seconds, I electrically stimulate the neuron, and we see that there's this robust calcium response in the connected neuropod cell. So at least in my in vitro system, I'm actually able to see that the efferent direction is able to modulate the activity of the neuropod cell. And so then what about the whole mouse? And so in our laboratory, we also are able to image in the gut. These data, they're still preliminary, and I'm showing you in the colon because the colon doesn't move quite as much as the small intestine, but I'm working on it. So in the colon, what I found is that these neuropod cells, when I'm just imaging spontaneous activity, they have these calcium oscillations. And this is interesting to me because what's just an observation I've made is if I just take them out and I culture them alone, the neuropod cells, there's no spontaneous activity. If I culture them with neurons, suddenly my culture is lighting up and there's spontaneous activity. So there's something about this connection that they're constantly communicating with each other. And we can see this in the mouse. And so now, what if I stimulate the vagus nerve while I'm recording activity in these neuropod cells? What I see is that, in fact, stimulation of the vagus nerve does seem to increase the amplitude and the frequency of these calcium oscillations that I see in the colon neuropod cells. And so the next big thing is, okay, well, what area could be actually feeding down? And so in order to do this, I used a rabies virus. So we use this rabies virus in order to do circuitry tracing in the gut. this virus has been modulated in 2 ways. It lacks the glycoprotein that's required for it to jump a synapse, and it expresses a Cre-GFP. So when I put this just in the lumen of the gut, I don't see any kind of synaptic tracing. It's because it can't jump, and it doesn't cross the epithelial layer. But now, if I put it in a Cre-dependent rabies G mouse, what happens is that the virus infects the cell. Since it's a Cre-dependent rabies G mouse, it then now expresses the glycoprotein required for it to jump onto the next synapse. And then I do see labeling. And now using this system, when it gets into the neuron, it can now jump another synapse and another synapse. And the cool thing about rabies, it's a retrograde virus. So it goes against the direction of activity. And so now I can go into the brain, and by counting my days, I can figure out, well, what regions are now efferently connected down into the small intestine onto these neuropod cells. And the first region I looked was, of course, in the brain stem, the dorsal motor nucleus of the vagus being the main efferent region in the brain stem. These reticular nuclei, they come kind of with the spinal cord innervation. They get labeled as well. We know that DRGs also innervate neuropod cells. But the next actual biggest region that I saw was actually the amygdala. And this was really surprising to me, because the hypothalamus is the area that we associate with food. But the amygdala would be this area that we associate with fear, anxiety, and stress. And we know that in chronic stress, it becomes hyperexcitable. And so now this picture is coming together of this circuit, and it's almost a loop. We sense nutrients. Say it's sugar. This is now communicated via glutamate to the nodus ganglia, goes into the brain stem, and then areas like the amygdala can now influence the dorsal motor nucleus of the vagus in order to then change how it is that these neuropod cells are actually sensing nutrients in the gut. And so this is how completing the circuit and figuring out how the efferent and the afferent are playing together is the focus of my future research. And so as everybody has mentioned, these things don't happen in a vacuum, and so I do have my acknowledgments of my lab, Diego Borquez's lab at Duke, which I work closely with, collaborators, including, of course, Polina, who made the flexible fiber optic, and my support at the NIDDK and at Duke. And with that, I am happy to take questions.
Time for questions?
Thank you. Excellent work. So I'm curious to understand and to know if you have any perspective of what could be the contribution of the 2 different parallel taste-like pathways from the gut and a superior processing of reward involving dopamine and more of a hedonic value of stimuli as well.
Yeah, it's a great question, and we also wonder this. So we rely on the tools that get developed, and one day I will find someone who says, yes, I have an activity-dependent synaptic tracing virus, and I will say, thank you. I want to know where these 2 pathways go. At the moment, we don't know that. What I think might be happening is that the sucralose might be using some kind of a bitter tasting pathway, potentially, because in the gut, sucralose is like, it really shouldn't be sweet, right? It's a fake compound. So we think that that might actually be doing some kind of either aversion or some other kind of learning, because we also know like medicinal plants, for instance, are also bitter. As far as the dopamine, so we know that the afferents do go into the hypothalamus where I expected and the reward pathways. But there's a postdoc in the lab that's actually looking at fat. And what she sees is that there's different types of fat, or there's different types of reward that are associated with fat sensing in the gut versus sugar sensing in the gut. So reward in the gut is not just reward is, reward is, reward is. It's actually like there's some kind of interplay about what types of reward, and we haven't quite figured out what that means for our overall behavior.
So if I may add,
Yeah.
you could also try, so just suggesting for us, quinine as well,
Yeah.
so like the bitter taste,
Yeah.
the aversive,
And
to see if it's going to be, for example,
the same between bitter and the sucralose. Yeah. Because there are bitter taste receptors in the gut as well. Mm-hmm. Yeah.
just curious if you had a chance to look at the left and right. I didn't hear anything about laterality, left vagus versus right vagus.
Yeah, so there's been some talk about left versus right. We record from the right vagus. I've done a little bit of left vagal work, and it also seems to be a sugar response, but we haven't actually been doing like at the same time to try and figure out if there's differences in reward or if there's differences in the pathway. Some people have said that there are, and there's some literature out there. Our sequencing data doesn't say that they're different, but I think that this might just be because we haven't quite gotten the right tool to be very specific about how this is working.
Hi, there's a question here, over here. To your right. Hi.
Hey.
So there has been some hypothesis going around that even a disease like Parkinson's is a prion disease that starts in the gut, which I would imagine that over time your body changes, especially like the pathway between your gut and your brain, how it ended up there, example. So do we have the tools to kind of monitor these changes over time in hoping that we can capture diseases like this early? And even just kind of like the food that you eat. For example, if you eat like, virgin, extra virgin olive oil versus if you just eat like butter all day, right? Is there like changes that affect your health that can be eventually translated to the neural system?
Yeah, so the prion diseases is interesting, and there has been some work that has shown that if you express the alpha-synuclein, you can actually see it jump from a neuropod cell onto a vagal neuron. However, there are questions about does it go brain to gut or does it go gut to brain? We know that it's capable of exchanging, but as far as early detection therapy, we're not entirely sure the direction of it. As far as the food that you eat, I mean, you also can't be in the gut and not mention the microbiome. So the food that we eat very much changes our microbes themselves, and we're just starting to learn about how it is that that's being sensed. And so we have a graduate student in the lab that's actually working on sensing of the microbial pattern proteins. And so what he's actually found is that a lot of this bacteria expresses something like flagellin. It's like for the little tail that wiggles on a bacterium. And what he's found is that if you knock out flagellin in colonic neuropod cells, the mouse eats more. And so what we're seeing is that, okay, the bacteria can't go to the supermarket. And when their levels get low, your flagellin signaling goes down. So if you fast a mouse, your flagellin goes down. And that could be communicating through these neuropod cells, hey, eat a little bit more during your meal. And then when the flagellin levels come back up, it's now signaling through this toll-like receptor 5, and it's like, okay, we're good. You don't have to eat quite as much. You can pay attention to some of the other things. So we think at least this is just one mechanism of how it might be that the microbes are communicating via these neuropod cells in order to change our feeding behavior. Whether that means you can eat butter all the time, I don't know yet. I would probably rotate in some olive oil in the meantime.
I think we have time for one more question.
Very nice presentation. And just on that point, do you know if these cells respond to the short-chain fatty acids produced by the microbiome? And I have a second question. The efferent connection from DMV, what would you see would be the advantage of having a system that controls the gain of these sensing neurons?
Yeah, so they do respond to the short-chain fatty acids, especially in the colon. For the efferent, actually, what I actually think is going on is there might be two mechanisms. One is an acute feedback loop that's happening, and this is very true with a lot of our senses. I use the eye a lot because we've studied it a lot. But our eyes, we're looking at something, it looks like it's still, it's not. Your eye is actually making tiny movements. It's because we only want to recognize change. And so what I think is that there's an acute feedback to then just like tune down quickly. Like I've sensed the first bite of cake. I don't need to sense like the 100 other bites of cake you're about to take now. But the DMV, I think what that is doing is now changing like the overall excitability. And so the DMV, they're these pacemaking neurons, right? So they have the ability to constantly be releasing something. And I think that when we get more into the circuitry, we might end up finding that that feedback might actually be almost like a saddle point. Because some people, when they're in like stress positions, they eat a ton more. Some people, they like stop eating completely. And it seems like two incredibly different, different phenotypes, but it could be because we have this constant interaction, this constant communication, and depending on what your genetics are or your previous learning, you might get pushed one side or the other.
Okay, thank you. Excellent job. Okay, our last speaker for this session is Dr. Jeanie Park. She is a nephrologist and associate professor of medicine at Emory University. And she's a deputy associate chief of staff of research at the Atlanta VA Hospital. Dr. Park is a physician scientist with clinical and research interests in autonomic regulation and neural control of blood pressure in disease states. Her research is funded by the NIH, the VA, Department of Defense, and some other sources. Please help me in welcoming Dr. Park.
I'd like to thank the organizers for inviting me to speak. It's been a fascinating session, and I've learned quite a lot since I've been here. I thought for this last session, I might give a little more of a clinical perspective on how there is a need to come up with new therapeutic modalities to improve autonomic function in our patients. And so I'm going to talk about the concept of leveraging interoception to improve sympathetic function in chronic disease states. And as a nephrologist and hypertension specialist, I see patients every day that have difficult to control hypertension. And I will tell you that the tools that we have right now to combat high blood pressure in our patients are inadequate. And particularly interventions that target the sympathetic nervous system. So we have diuretics that we use vasodilators, we use inhibitors of the renin-angiotensin system. And you might say, oh, well, you have beta blockers and alpha blockers and you have clonidine. But these are medications that, yes, theoretically combat sympathetic overactivation, but their use is limited by side effects. And in fact, when you give beta blockers or alpha blockers, you have this reflex increase in sympathetic activity. So you're not actually modulating sympathetic tone overall. And so there is a critical need to develop new therapeutic modalities, such as through neuromodulation, to improve blood pressure, autonomic function, and long-term clinical outcomes. And so that is the perspective that I have for the work that we do in our lab. And so we've seen several definitions already of interoception, but we can think about interoception as the sensing of the internal body sensations. And so this is in contrast to exteroception, which is how we sense and interact with the external environment. Interoception is the sensing of the internal landscape. And interoception is critical for maintaining homeostasis. And we know that there are a number of diseases that are characterized by aberrancies in several interoceptive pathways, and these can then lead to long-term derangements in homeostasis and chronic disease. So there are multiple components of interoception, as has already been described in some previous talks, but we have afferent sensory or bottom-up pathways that are largely unconscious. So for example, the Darrow reflex is mediated by sensory nerve endings that reside in the carotid sinus and detect changes in arterial blood pressure and will modulate blood pressure or sympathetic activity in response to those afferent inputs. And that is an unconscious process. And then there are also top-down or conscious pathways that regulate autonomic function as well. So for example, heartbeats and breathing are automatic processes that are usually unconscious, but they can be brought into conscious awareness and can also modulate sympathetic tone. And so these interoceptive inputs maintain homeostasis, and they do so through covert reflexes. So I mentioned the Darrow reflex, which makes automatic adjustments to sympathetic output based on changes in blood pressure. there's also an effect on motivational drivers. So for example, when you have gastric emptying, the stretch receptors in your stomach become unloaded, and that creates this feeling of hunger, which then drives the behavior of eating. And there are other bodily sensations as well that are driven by these pathways. So the sensation of breathlessness, for example, hunger, pain, etc. And so what are the major regulators of the sympathetic nervous system? So the main effect of sympathetic nerve activation is that there's a release of norepinephrine and other neurotransmitters from sympathetic nerve terminals. And this then results in an increase in cardiac output and vasoconstriction that increases blood pressure. And so I've already mentioned the Darrow reflex, which is one of the peripheral regulators of the sympathetic nervous system. So the arterial Darrow reflex receptors are nerve endings that reside in the aortic arch and the carotid sinus and modulate sympathetic output on a beat-to-beat basis based on changes in blood pressure. The cardiopulmonary baroreceptors are sensory nerve endings that reside in the atria and in the lungs that sense changes in blood volume and then will modulate sympathetic output based on changes in volume status. And so these peripheral afferent or interoceptive pathways occur automatically and are largely unconscious. And then there are also central or top-down or conscious pathways that also can have a modulatory effect on sympathetic output, such as, for example, the relaxation effect, or the perceived exertion of exercise, for example, will increase sympathetic activity even without physical activity. And so we know that the sympathetic nervous system is important for moment-to-moment control of blood pressure, but it turns out that the sympathetic nervous system also plays a major role in long-term blood pressure regulation. And when there are derangements of sympathetic control, overactivation of the sympathetic nervous system, or dysregulation of the sympathetic nervous system, these are processes that are independent risk factors for cardiovascular disease and mortality. And we know that there are many diseases that are characterized by chronic overactivation of the sympathetic nervous system. So this includes hypertension, heart failure, vascular disease, obesity, chronic kidney disease, and post-traumatic stress disorder. So if you look at this list, and this is not a complete list at all, these are very, very common health conditions that are characterized by derangements in sympathetic regulation, for which we really do not have good treatment options, particularly to target autonomic dysfunction. And I'm going to talk more about chronic kidney disease and post-traumatic stress disorder in general because these are patients that I take care of. So I just want to give you a little background on chronic kidney disease. I don't know if everyone knows quite how prevalent this disease is, but about 37 million US adults have chronic kidney disease. So that is more than the number of US adults that have diabetes. And it's about 15% of the US adult population. And there are 850 million people with chronic kidney disease worldwide. So this is a very highly prevalent disease. And these patients, by virtue of having a decrease in kidney function, they're at significantly higher risk of developing cardiovascular disease and mortality, and they have high blood pressure that is more difficult to control. So we have many epidemiologic studies that have shown this early mortality risk. I'm just showing one here in which you can see that as kidney function declines, measured as estimated glomerular filtration rate, the hazard ratio for both all-cause mortality as well as cardiovascular mortality goes up. And one major mechanism that underlies increased risk for cardiovascular disease, mortality, sudden death, and difficult to control hypertension is chronic overactivation of the sympathetic nervous system. So here on the left panel are some neurograms of muscle sympathetic nerve activity in 4 patients with chronic kidney disease compared to 4 controls that are matched for age and comorbidity and BMI. And you can see that those with chronic kidney disease, hopefully you can see that the number of bursts of sympathetic activity are much higher in the CKD patients compared to the controls. And in fact, it's been shown that the lower your kidney function, the higher your resting sympathetic nerve activity. So not only is resting sympathetic activity elevated in chronic kidney disease, but the reactivity of the sympathetic nervous system is also augmented. So here we showed that during static hand grip exercise, which is a sympathoexcitatory maneuver, on the left, in the black dots, that's the chronic kidney disease. Patients have this augmented mean arterial pressure response during hand grip, and concomitantly, they have this exaggerated increase in sympathetic nerve activity that occurs during hand grip exercise. Okay, so what about post-traumatic stress disorder? So you might be asking, well, why is a nephrologist interested in PTSD? So I have been a physician at the VA for over 10 years now, and many of my patients have comorbid PTSD, and it has a definite impact on the way that, that we manage our patients. And so I became interested in the pathophysiology behind this disorder, and it turns out that PTSD is independently associated with increased cardiovascular risk as well as overactivation of the sympathetic nervous system. So PTSD is characterized by persistent mental and emotional stress that develops after severe psychological shock. It's quite prevalent amongst military veterans, so 12 to 30%. So it's about 12% in Gulf War era veterans, 30% in Vietnam War veterans, and about 20% in post 9/11 veterans. And so, by the way, there were over 20 million people that were deployed for these conflicts in Iraq and Afghanistan, and 20% of them have PTSD. So there is a huge number of young people now that have PTSD and now have increased cardiovascular risk, increased risk of hypertension due to PTSD. So this is a condition that's also highly prevalent amongst the general population. So the risk for, the lifetime risk is about 7 to 12%. And again, PTSD, we know, is independently associated with an increased risk of hypertension and cardiovascular disease. And this has been shown in multiple large epidemiologic studies that have been well controlled for other potential confounding comorbidities. And the risk appears to be about 2-fold in terms of developing cardiovascular disease. And these are data from the U.S. National Comorbidity Survey that shows that patients with PTSD, with and without depression, are at about a 3-fold greater risk for developing hypertension. So interestingly, when we looked at this literature, no one had really previously done direct measurements of sympathetic activity and regulation in PTSD, although there was an assumption that the autonomic balance is off in PTSD. So what we did was we measured changes in sympathetic nerve activity during sympathoexcitation induced by mental stress. And so the first stress that we used is virtual reality combat exposure. And this was in combat veterans that had combat-related PTSD. And so as you might expect, those with PTSD, shown in the black circles, they have this exaggerated increase in sympathetic nerve activity during virtual reality combat exposure. But what was interesting and not expected is that this was not confined just to mental stress that was related to PTSD symptoms, but also to other types of mental stress as well. So here during mental arithmetic, we observed this greater increase in sympathetic activity compared to veterans without PTSD. And so this is important because we know that exaggerated sympathetic reactivity is associated with an increased risk of hypertension and cardiovascular disease. We also examined the regulation of the sympathetic nervous system by the arterial barrow reflex. And we use the modified Oxford technique where we give a bolus of nitroprusside, which then leads to a decrease in blood pressure, and there should be a reflex increase in muscle sympathetic nerve activity that is driven by the arterial reflex, with a reflex increase in heart rate and a reduction in an RR interval. And then we give a bolus of phenylephrine, which increases the blood pressure, and thereby you should see a reflex dampening of sympathetic nerve activity with a reduction in heart rate and an increase in RR interval. And we can quantify the sensitivity of the Darrow reflex by examining the slope of the relationship between diastolic blood pressure changes and muscle sympathetic nerve activity, as shown in that figure on the right. And so the steeper that relationship, the greater the Darrow reflex sensitivity. And conversely, the flatter that relationship, the more blunted the sympathetic Darrow reflex sensitivity is. And so when we did this in PTSD patients, what we found was, in fact, PTSD patients have a blunted sympathetic Darrow reflex sensitivity. So their slope is shown there in the solid line, whereas veterans without PTSD are shown in the dashed line. And this is important because this may be contributing to sympathetic overactivation, but impaired arterial Darrow reflex sensitivity itself is also an independent risk factor for developing hypertension and cardiovascular disease. And so why is this important? Why is sympathetic overactivation such a major clinical problem? Well, so sympathetic activation increases blood pressure. So there's a release of norepinephrine, as we mentioned, that causes vasoconstriction and increases blood pressure. But there are a number of other deleterious consequences of overactivation of the sympathetic nervous system that increases mortality risk and risk of cardiovascular disease. So for example, to the heart, sympathetic activation contributes to left ventricular hypertrophy. It increases arrhythmogenicity, which increases the risk of sudden cardiac death, which is a major cause of death, actually, in patients with chronic kidney disease. To the muscles, it will contribute to insulin resistance. To blood vessels, causes vascular stiffness, accelerates atherosclerosis, causes vascular inflammation. And to the kidneys will increase sodium retention, which further increases blood pressure, activates the renin-angiotensin system, reduces kidney blood flow, and accelerates glomerulosclerosis and decline of kidney function. And again, our current therapies for sympathetic overactivation are limited. So again, we're limited to largely pharmacologic agents, peripheral sympatholytics like beta blockers and alpha blockers, and central sympatholytics like clonidine. But again, these drugs do not protect against cardiovascular risk as well as other antihypertensive medications. And they often have intolerable side effects, especially in older patients, that really limit their use. And so again, there's a critical need to develop new treatment strategies that target sympathetic overactivity and dysregulation. And so can we leverage any of these pathways, these interoceptive pathways that regulate sympathetic activity to intervene on autonomic function in a way that is safe and well tolerated? So again, interoception refers to the sensing of the internal state of the body that provides the afferent channel of the interplay between body and the brain that allows homeostasis through covert reflexes, for example, the Darrow reflex, motivation drivers, and explicit body sensations. And so our thought here was, can we leverage the interoception of breathing in order to improve autonomic function in patients that have overactivation of the sympathetic nervous system? So the rationale here is that we've actually known for quite some time that slow breathing reduces blood pressure. And the purported mechanism is that when you reduce your respiratory rate, then you increase your tidal volume. And so you do that in order to maintain the same minute ventilation. And so when there is an increase in tidal volume, this then activates the cardiopulmonary stretch receptors, which then leads to a reflex decrease in sympathetic activity and a reduction in blood pressure. In addition, however, there is also a conscious awareness of breathing. Again, breathing is an automatic process, but can be brought into conscious awareness that may also modulate sympathetic activation. So we did a very simple experiment in which we simply measure the change in sympathetic activity during slow breathing. And we used this device, the respirate device, which is shown there in the middle. This device has a respiratory belt that goes around the upper abdomen, so it monitors breathing rate. And then it has headphones where it delivers musical tones to effortlessly reduce the respiratory rate down to about 5 to 6 breaths per minute. So the normal physiologic respiratory rate is between 12 to 20. So this is a substantial reduction in the breathing rate. And so then the participant is then synchronizing their breathing with these guiding tones. And so this device, the respirate device, it is currently FDA approved for, and I'm quoting the indications for relaxation and the adjunctive treatment of hypertension. And again, slow breathing, we think, could engage in components of interoception, including the afferent activation of these pulmonary stretch receptors, as well as increasing the awareness of the bodily sensation of breathing. And so what we found, and I just want to give a shout out to Dr. Ida Fanqui, who was previously a postdoc in the lab and is now an assistant professor here at the University of Minnesota. She led these studies that we did in post-traumatic stress disease. And what she showed is that during device-guided slow breathing, there is a significant reduction in systolic blood pressure compared to sham breathing. So our control intervention was the same device, but it was programmed to guide breathing at a normal respiratory rate of 12. And then concomitant with that, we observed this difference in the sympathetic response, where those that were breathing slowly had a reduction in sympathetic activity. And Ida also then did the modified Oxford technique before and after slow breathing versus sham, and she found that after device-guided slow breathing, there is an improvement in the sympathetic baroreflex sensitivity compared to sham, which actually there was some worsening in baroreflex sensitivity with sham breathing. And so based on these acute studies, we then did a clinical trial in which we wanted to investigate whether there could be a sustained sympathoinhibitory effect to device-guided slow breathing. So patients with PTSD were randomized to 8 weeks of slow breathing with the respirate versus 8 weeks of sham control. And what we found was that there was no change in resting sympathetic activity, but there was a difference in the sympathetic response to mental stress, such that those that did 8 weeks of slow breathing did have an amelioration in their sympathetic reactivity to mental stress. So another breathing intervention that we have had interest in is mindfulness meditation. So mindfulness meditation often has a component of increasing breathing awareness. And so in these studies, we enrolled untrained African-American men with chronic kidney disease, and they underwent either 14 minutes of mindfulness meditation with an emphasis on breathing awareness, and this was compared to a health education recording as the control. And what we found is that after about 6 minutes, there was a reduction in sympathetic nerve activity in those that were randomized to the mindfulness meditation with breathing awareness versus health education recording. And there was a very small but significant reduction in mean arterial pressure as well as heart rate. And what was also an interesting observation from this study is that mindfulness does not necessarily manipulate the breathing rate, but there was a natural reduction in the respiratory rate in those that underwent mindfulness meditation. And so we did another series of experiments in which we had the participants just decrease respiratory rate without the mindfulness component. And what we found is that we called that intervention controlled breathing. So with controlled breathing, there is no effect on sympathetic activity like we saw with mindfulness. So it appears that slow breathing alone was not sufficient to lower sympathetic tone, but that mindfulness, that conscious component seem to be necessary for the physiologic effect that we observed in chronic kidney disease. And so mindfulness, again, is leveraging both the peripheral pathway with a reduction in respiratory rate that increases the cardiopulmonary baroreflex and reduces sympathetic activity, as well as a feed-forward mechanism that is mediated by an increase in breathing awareness. So to summarize, manipulation of respiratory rate and increasing breathing interoception may have acute sympatholytic effects, may also improve baroreflex function, and might have sustained sympathoinhibitory effects in chronic disease states that are characterized by overactivation of the sympathetic nervous system. And this is just an example of thinking outside the box on how we can come up with different modalities to improve autonomic control in patients with chronic diseases. And these are not uncommon chronic diseases. And so some of the questions that we have going forward, what are the most effective therapeutic modalities that leverage interoception to reduce sympathetic activation? And do these therapies lead to improved clinical outcomes? And I do want to say that we have ongoing studies now where we're trying to optimize the sympatholytic effects of mindfulness by adding vagus nerve stimulation. And those studies are now ongoing, funded by NCCIH. Thank you, Wen. And I'll stop there, and I'm happy to take questions.
Okay, we'll take a couple questions.
I've got one. Hi. In some of the plots that you showed kind of time differences and how things like spread apart over time. When do they come back together? So for example, when you had the 14-minute video, did you continue to record after those 14 minutes and see when the blood pressure came back up? Or when you had your 8-week trial, did you find that as people did it more and had more practice at it, that those effects lasted longer?
Yeah, that's a really good question, because obviously it's not going to be clinically important unless we can really observe a sustained effect. And so we do have recovery data of after mindfulness, and these were in untrained individuals, and so there was really pretty much almost instantaneously they went back to their state, their normal, the sympathetic activity went back up. Same is true actually with device-guided slow breathing as well. After the training effect, we did see some amelioration in the sympathetic response, but we don't see a reduction in resting sympathetic activity. And I think one of the issues with that is that unless you're actually doing the practice, you're breathing at your normal respiratory rate. And so right now it really seems like the acute effect is much more prominent than a sustained effect. But I think that this is something, though, that we need to work on in terms of how do we optimize this. Is there a way to retrain breathing, for example, so that people naturally breathe at lower rates or naturally have a higher kind of mindfulness type of awareness at all times? But yes, that is one of the limitations.
Okay. Hi, Jeanie. Very nice talk, and thank you very much. I have actually two questions. One is more related to your talk, but the other one is actually just a probably general comment about the whole session. So regarding to your talk, your data of showing mindfulness, breathing versus slow breathing that has differential effect is quite fascinating. In fact, Vidal Zayden at UCSD had done very similar study, but in pain patient, and he neuroimaged their brain activity and found out it's activating different circuits in the brain. So that might be of interest to you. And I wonder if there's some connections here. I'm really curious about what is different between a mindful breathing versus a slow breathing. So
that's actually a really good question. I've been thinking about that quite a bit, because if you decrease respiratory rate enough, though, like down to 5 to 6 breaths per minute, even without that conscious component, the mindful component, you can see sympathetic activity go down. So it's seems like it might actually be the degree of reduction in the respiratory rate, but the conscious component definitely has an additive effect. And I think it's much more feasible to think somebody's going to have a respiratory rate of 12 that they can sustain versus a respiratory rate of 5 to 6 that they can, continue to sustain. So yes, I think that conscious component is totally necessary when your rates are like, just a little bit low, like 10 to 12 or so. It enhances.
What did you ask them to do when they were doing mindfulness, actually, exactly? I'm sure it was in your protocol, I can't remember now.
Oh, yeah, so they are listening to a breathing awareness meditation. So that was in the initial acute studies. But now they're actually doing, we're doing a chronic study now. They're doing MBSR, so they're doing the regular mindfulness-based stress reduction.
My second comment is about the whole definition of interoception. So I was explaining to John during the morning break that actually following the NIH workshop in 2019 on interoception, we published the paper which Manda, you referenced earlier. In that paper, we actually intentionally changed the traditional definition of interoception to be including not just the sensing or the efferent part, but it also includes the entire integration and processing part in the brain, as well as the efferent part. And that's really, I know people are always trying to just want to focus on the perception part, and I explained it a little bit earlier to John that I think that there's a very purposeful action here of why we want to include the regulation, which is actually encompassing the entire autonomic nervous system as well. So the definition of interoception is, at least as at the NIH, how we have perceived it is what we had thought about as the sensory part, and then the entire brain processing, as well as the entire autonomic nervous system. Just because if we don't include them, then you get into this linguistic about interoception regulating sympathetic activity, which doesn't make sense. It's the same pathway. It's actually, we're all looking at the same thing, and it makes research really challenging. And also when you measure a lot of what we call interoception, you're actually measuring sympathetic or parasympathetic activity. So it doesn't make sense to think as if they're two different things. But instead, we basically said maybe you're reading the output, the top-down regulation or processing. And also they do. And then also the regulation also regulate the internal signal that goes back in the brain. So it's sort of a perpetual circuits to some degree. So it's really important, I think, for the field to not try to separate them in the way that make as if they're two different things. I think that will make your research really challenging later, and it's very hard to explain and come up with theories too.
Thank you.